What is a breakthrough infection? Why is COVID-19 vaccine breakthrough occurring? And how does its breakthrough frequency compare to that of other viruses and vaccines? Hi, and welcome to Microbial Minutes. This is the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Ashley Hagan, Science Communications Specialist for ASM. And today we're talking about SARS-CoV-2 variants, vaccines, and breakthrough infections. Amidst encouraging real-world data about the effectiveness of COVID-19 vaccines, and the CDC's announcement last month that fully vaccinated individuals can engage in indoor and outdoor activities without a mask, reports of fully vaccinated people becoming infected with SARS-CoV-2 have raised questions about the frequency and cause of breakthrough infection. Why are some people who are fully vaccinated still testing positive for the virus? We're going to take a look at a few preliminary studies in which researchers are beginning to evaluate trends in case characteristics and sequencing data from breakthrough SARS-CoV-2 infections. But first, we need to address a couple of foundational concepts. Number one, a breakthrough infection occurs when an individual becomes infected with a pathogen that they have been fully vaccinated against. It takes about 2 weeks after vaccination for antibodies to develop and be protective against future disease. Therefore, a person is not considered fully vaccinated until 2 weeks after completing the recommended number of doses for the vaccine they were administered. And this is true for most vaccines, not just COVID-19, because the delay is a factor of the host's immune system and not specific to the antigen being introduced by vaccination. Number two, it's important to recognize that unless a vaccine produces sterilizing immunity, which completely prevents a disease-causing pathogen from establishing infection with 100% efficacy, small percentages of breakthrough cases will occur and are actually expected. For example, data suggests that mRNA COVID-19 vaccines are over 90% effective at preventing SARS-CoV-2 infection. The chickenpox vaccine is 85% effective at preventing varicella-zoster infection. And the MMR vaccine is 88% effective at preventing mumps. Number three, there are a number of reasons that breakthrough infections may occur, including a weak or ineffective immune response to vaccination, decreased immunity over time, and/or evolution of the virus. The fact that SARS-CoV-2 is mutating is not surprising. All viruses mutate, but the virus's mutation landscape is an important factor to consider when it comes to the idea of immune escape or evasion. On May 25, 2021, the CDC published some preliminary results of national surveillance efforts to investigate SARS-CoV-2 infections among people who are fully vaccinated. A total of 10,262 breakthrough cases were reported between January 1st and April 30th of 2021 from 46 U.S. states and territories. Considering that at that time 101 million people had been fully vaccinated in the U.S., these numbers point to a low breakthrough rate of approximately 0.01%. But the actual frequency of breakthrough infection is difficult to ascertain because it is unlikely that all cases are being diagnosed or reported, especially if symptoms are mild to asymptomatic. So what was the outcome of these cases, and who was most susceptible to breakthrough infection? According to the report, 2,725, or 27% of the aforementioned cases, were asymptomatic. 995, or 10%, required hospitalization, and 160, or 2%, resulted in death. It is presumable that the majority of cases developed mild to moderate symptoms not requiring hospitalization. The median age range of the patients who died was 82 years, and 18% of the deaths were asymptomatic or attributed to a cause unrelated to COVID-19. Representative sequence data was available for only 555, or 5%, of reported cases, and revealed that variants of concern, or VOCs, were responsible for 64% of those cases. The majority of these were identified as B117 or B1429. Previous studies have indicated that in the lab, antibody neutralization is either similar or slightly reduced against certain SARS-CoV-2 variants relative to wild type. For example, a 2 to 7-fold reduction against B1427 and B1429, a 4 to 5-fold reduction against P1 or gamma, and more significant observations of 5 to 40-fold reduction against B1351 or beta have been reported. But the question remains, are COVID-19 vaccines less effective against these variants in the real world? If so, one would expect the proportion of VOCs to be higher amongst vaccinated individuals than unvaccinated ones. During the 2 weeks of March 28th through April 10th, 2021, the proportion of variants of concern circulating in the U.S. accounted for 70% of the weighted estimates submitted to the CDC's National Genomic Surveillance Program, suggesting that SARS-CoV-2 variants were not significantly enriched in breakthrough cases. However, a preprint posted on the same day identified and sequenced 20 breakthrough cases in Washington state and found that all 20 could be attributed to variants of concern. When compared to sequencing data from the control group, 5,174 samples collected from non-vaccinated individuals in the same area during the same time period, researchers found that VOCs were significantly enriched in breakthrough cases. None of these patients required hospitalization, and 15 reported symptoms of COVID-19. Here you can see a visual representation of the percentages of variants that were detected in the breakthrough cases, as well as the circulating population. 8 breakthrough cases were identified as B117 or alpha, 1 as B1351 or beta, 2 as B1427, 8 as B1429, and 1 as P1 or gamma. Next, researchers began to compare the occurrence of individual mutations in the spike protein to see if there was any detectable enrichment amongst breakthrough cases. Spike is the main protein used as the target of current COVID-19 vaccines. The S protein is 1,273 amino acids in size, and it is the portion of the virus that recognizes and binds to host cellular receptors and mediates viral entry. And SARS-CoV-2 is unable to infect host cells without it. Because of this, mutations in the S gene, particularly those that affect portions of the protein that are critical for pathogenesis and normal function, such as the receptor binding domain or furin cleavage site, or those that cause conformational changes to the spike protein, are of greatest interest. If the adopted changes are not recognized by first-wave antibodies, these mutations may provide an avenue for the virus to escape from immunity and offer a plausible explanation for COVID-19 vaccine breakthrough infections. In this study, a single tryptophan to leucine substitution at position 258 was enriched 15.22-fold in the breakthrough infections. Although the mutation was only present in 2 of the 20 cases, it is located in an antigen supersite, a vulnerable region in the N-terminal domain of the SARS-CoV-2 spike protein that is targeted by neutralizing antibodies. Additional research suggests that mutations in this region may be associated with immune evasion. Another preprint posted on May 31, 2021, which analyzed over 1.3 million SARS-CoV-2 genomes from 178 countries and performed whole genome sequencing from 53 COVID-19 patients, 20 of which were breakthrough infections, found that deletions in the NTD antigenic supersite were highly enriched in surges of COVID-19 that occurred in different parts of the world, including India and Chile, and appeared in breakthrough infections. 92 unique mutations were identified in this study. 28 of those were deletion mutations, and all of the deletions that were identified in the spike protein occurred in its N-terminal domain. It is unknown whether these deletions were present at the time of infection or evolved under selective pressure of vaccine-induced immunity. But researchers suggested further epidemiologic monitoring of the 4 spike protein variants harboring 1 or more deletions, shown here, to determine whether or not they impact vaccine effectiveness in the coming months. One of these variants, which is represented in this figure in green, contains deletions at position 156 through 164, suggesting that the prominent F157R158 deletion detected in surges in India might actually be part of this larger deletion-prone region in the NTD antigenic supersite. Here are a few key takeaways from today's session about breakthrough COVID-19 infections. Unless a vaccine is 100% effective at preventing infection, a small percentage of breakthrough cases are expected and not overly concerning. At this time, the frequency of COVID-19 vaccine breakthrough is very low, about 0.01%, which means COVID-19 vaccines are highly effective at preventing COVID-19, particularly when it comes to severe disease. There are a number of reasons that breakthrough infections may occur, including a weak or ineffective immune response to vaccination, decreased immunity over time, and/or evolution of the virus. Evidence suggests that certain mutations may be enriched in breakthrough SARS-CoV-2 infections, and deletions in the NTD antigenic supersite are of particular interest and require further investigation. So that's the latest on what's hot in the microbial sciences. We will continue to provide updates as new information and data become available. Don't forget to subscribe to ASM's YouTube channel and ring the bell for notifications every time we have a Microbial Minutes update. I'm Ashley Hagen. I'd like to thank you for listening and thank Ray Ortega for production. I'll talk to you next time.